Hello dear students, myself Suchita Bidve and I am working as Assistant Professor in Department of Botany, KTHM College, Nasik. Students, for your S.Y.B.Sc semester 2nd, I am going to deal with subject Botany paper 2nd Plant Biotechnology and our course number is BO242 Plant Biotechnology. Students, this theory paper is of 2 credits. First credit is for plant biotechnology and second credit is for advances in plant biotechnology, that is genetic engineering. Student, this course is totally formed with the 30 lectures and each credit is of 15 lecture. So for this semester I am going to deal with credit second, and from this video we are going to start with our credit second, that is plant genetic engineering. Students, plant genetic engineering is a very recent modern concept and that will be useful for crop improvement. This science is mainly derived to develop new varieties of plant which are economically important, and mainly this involves the preparation of hybrid plants which are tolerant to biotic and abiotic stresses. These are high yielding, good nutritive values, etc. can be developed with the help of plant genetic engineering. Students, this science is mainly based upon the modification within the gene and cloning of specific fragment of DNA, that is gene, into another economically important plant for specific required trait. Basically, genetic engineering is a direct manipulation of genes, genetic makeup, that is DNA, genetic constitution of an organism as desired using different biotechnological tool. Today, this tool we can apply mainly for the tremendous changes in the agricultural sector, for health, for food, for environment to get a desired valuable product for human beings. This biotechnology is a science. This genetic engineering is a direct manipulation of genes, genetic makeup, genetic constitution of an organism as a desired using different biotechnological tools. And here the different biotechnological tools used are specifically cutting and joining tools of DNA like restriction enzymes, ligases enzyme. Basically, the plasmid vectors are required for these biotechnological operations, and PCR is a new recent technique is used for the cloning purposes. Mainly, genetic engineering is a transfer of specific segment of DNA within and across the specific boundaries to produce novel organism as per goal. Specifically, remember one thing, here we are selecting a specific gene from one organism and we are inserting that specific gene within the another organism in order to achieve a goal of a novel organism with a specific characters. Okay, for example, if we are interested to develop a plant variety with salt resistance, we know soil may be having high content of soil, within that high content of salt, and within that type of soil there is no proper growth of plant. So if we insert a salt tolerant gene into economically important varieties of plant, then we can acquire or we can produce a plant variety with a salt resistance capacity. If we want a scientific definition of genetic engineering is artificial manipulation, modification, and recombination of DNA or nucleic acid molecules in order to modify an organism or its population. Mainly, this genetic engineering is nothing but the recombinant DNA technology or gene cloning. This recombinant DNA technology having its some valuable economically important creations like BT plants like cotton, brinjal, banana, potato and tomato is possible tough genetic due to tough genetic engineering tools. Likewise, we are also familiar with golden rice, the rice rich in vitamin A precursor, flavor sable tomato with high shelf life. Then along with that, we are also knowing the creations like Dolly Sheep, Test Tube Baby is the success of gene cloning. Similarly, in vitro fertilization, artificial insemination in livestock is only possible due to genetic engineering, and the organisms which are produced by this genetic engineering tool are also known as genetically modified organisms. These genetically modified or genetic engineering are having their lot many applications in the gene therapy, in enzyme engineering, in enzyme technologies. We can increase a photosynthetic efficiency of plant by using genetic engineering also. This field, this genetic engineering is also helpful in medicinal field for production of vaccine in order to develop a unique drug delivery system, as well as this can be useful for development of biosensors, and genetic engineering are also having their recent applications in nanotechnology also. When we talk about genetic engineering or recombinant DNA technology, this recombinant DNA technology is mainly based upon three important tools of biotechnology. That is, first very important tool is restriction enzymes, which are used as a DNA cutter. Then next important thing is DNA ligases, which are used to join different fragments of DNA. And the most important thing which is important for cloning purposes is plasmid vectors. Now we'll see one by one each tool of genetic engineering. First we'll start with restriction enzymes. Restriction enzymes are also known as molecular scissors. These are cutting tools of DNA. These are basically enzymes who having capacity to cut the DNA at specific sequence only, and therefore these are also known as sequence specific cutters of the DNA. These restriction enzymes are basically originated within the bacteria naturally. These are present in bacteria as a defense mechanism, specifically to protect themselves from the foreign DNA like bacteriophages. Bacteriophages, these are the viruses which are infecting the bacteria. These are these restriction enzymes are mainly evolved in bacteria in order to cut the foreign DNA, and these enzymes are used in biotechnological purposes also. These enzymes, these restriction endonucleases or exonucleases are specific, and these are recognizing 4 to 10 base pairs of DNA. They are having ability to cut the DNA at sugar phosphate backbone, and they are having capacity to produce two different fragments of DNA. These restriction enzymes are of different type: endonucleases depending and exonucleases, and again depending upon site of cleavage they are having a there are different types of restriction endonucleases. We'll see one of the example from the restriction endonucleases. Here the enzyme hinge 3. H3, this is named after the bacteria Haemophilus influenza. It is derived from Haemophilus influenza. Now, in diagram we can see a specific sequence AAGCTT. Okay, it is present and 5 prime on 5 prime to 3 prime end and from 3 prime to 5 prime end between the 2A, that is 2 adenine, between the 2 adenine nucleotide, this hinge 3 is able to cut. For the identification of cutting site, it requires a specific sequence, that is AAGCTT, and it is able to cut between 2A. So it is producing two unequal ends from 5 prime to 3 prime end and 3 prime to 5 prime end. Likewise, there is another restriction enzyme EcoR1. Again, it is originated from bacteria Escherichia coli and it recognizes a sequence GAATTC and it cuts specifically between G and A in order to produce the fragment of DNA, the cut site of the DNA. Now, these cut sites of the DNA can be joined with the help of DNA ligase enzyme. Ligases are having ability to join the cut ends of DNA. By using energy, that is ATP, it can catalyzes the joining of phosphate group of 5 prime end of one DNA and hydroxyl group of 3 prime end of another DNA molecule, so that it leads to sealing of sugar phosphate backbone of the DNA molecule and it produces a recombinant DNA. Basically, the T4 DNA ligases are used to generally used in genetic engineering operations. See here, there is one example we can see here. There is cut cut sticky ends of DNA which are produced by EcoR1 enzyme that can be uh, joined by using ligases enzyme. It is having capacity to join the or to prepare a sugar phosphate backbone of DNA molecule which leads to formation of recombinant DNA molecule. Likewise, the last tool of the genetic engineering is a vector. Vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell where it can be replicated or expressed. Generally, vector used in a genetic engineering purposes are DNA and these are extra chromosomal or extranuclear genetic material, extra chromosomal genetic material which are generally found in lower organism like bacteria or viruses. There are some types of vector. Commonly used vector are plasmid, cosmid, lambda phage vectors. Vector can carry foreign DNA molecule from one cell to another cell and it has a capacity to replicate inside the host cell. Plasmids generally used vectors are will initially we'll see silent features of vector. A vector should has its own replicon. It should have a original of replication. It should have a presence of some marker gene to differentiate transform and non-transform cells. It should have a presence of unique cleavage site with the marker gene and it should have presence of specific control elements like promoter, terminator, ribosome binding sites, etc. Now, when we talk about the vectors, vectors are generally plasmid vectors are preferentially used as in a genetic engineering tool. Plasmids are simply transferable extra chromosomal DNA molecule which are capable of autonomous replication. They are double stranded, mostly circular in nature. It is usually occurs naturally in bacteria and is sometimes found in eukaryotic organisms also. Mainly, now we'll see some used examples of vectors. First, we'll see PBr322. This PBr, P stands for plasmid and here Br stands for name of scientist. Here B for Bolivar and R for Rhodys. And this 322 is a number. This PBr3 is used as a cloning vector and this has capacity to present in 15 copies per cell. It is a first artificial cloning vector to be constructed within the laboratory and used most widely for all the cloning purposes till today. It is 4.36 kilobase pair double-stranded cloning vector. This plasmid contains 20 unique sites for recognition of restriction enzymes. It is derived from E. coli plasmid col E1 and it has a two selectable marker ampicillin resistance gene and tetracycline resistance gene. There is another example very commonly used vector is PUP19. Okay, it is again a cloning vector and this PUC that is plasmid from University of California 19. It is originally derived from E. coli 1, E. coli and it has a origin or main origin sequences are derived from plasmid col E1. It has ampicillin resistance gene as a selectable marker, laxate gene as a screenable marker, and it has a presence of 54 unique restriction site. This is the size of plasmid is 2.6 kilobase pair and till it is commonly used in all type of cloning purposes. The next type of plasmid are TI plasmid. These TI plasmid are large circular DNA molecule and their size is always ranging between from 200 kb in length and with mostly about 1.2 into 10 raised to 8 kilobase pair. They exist in a bacterial cell as an independent replicating genetic unit. These TI plasmid are mainly derived or based on plasmids present in a bacteria Agrobacterium tumefaciens. These are the plasmid which are having capacity to carry gene into the plant cell. DNA plasmid have a major region for virulence, origin of replication, conjugations, oncocity, and catabolism of opines. Genetic organization of this region is shown in a figure and plasmid are used for the cloning of larger gene. Generally, these plasmid are having its own origin of replication. These are having virulence gene. These are the group of genes which are required for transfer of tDNA. They are having tDNA region. tDNA, it is a transferable DNA which is having presence of left border and right border and between this there is presence of auxin and cytokinin synthesizing gene, okay, which are also known as oncogene. And there are again there is presence of genes for opine catabolism. This is structure of TI plasmid. So student, in today's session we have seen concept of genetic engineering and tools of genetic engineering. In next lecture we'll see what is exactly cloning, what is procedure of cloning, and how we can do cloning and what are the applications of cloning. If you have, these are some references which I have used for your lecture, and if you have any query you can mail me on my mail or you can discuss in our Google Classroom. Thank you.